Hi everyone, thanks for choosing the proteomics unit. Over the course of the next 6 contact hours, we're going to be introducing concepts important to proteomics and the analysis of omics data. That's going to include fundamental concepts in mass spectrometry, as well as concepts specific to proteomics and the acquisition of high quality data, and then the analysis of that data. So this first section is going to be an introduction. Primarily we're going to be going through the fundamentals of acquiring and understanding proteomics data. So proteomics and omics in general is really asking the question of, in a biological sample, what is that sample composed of? What is the sequence of the genes which are present in that sample? How much and what are the sequences of RNA molecules present in that sample? How many and what types of proteins are present in that sample? And the same for small molecules. As you can see in this graph in the top right, there are radically different amounts of different biomolecules present in a cell. There's a vanishingly small amount of DNA, there's some RNA, there's a lot of protein, and there's even more small molecules present there. And the technologies for accessing these different types of biomolecules present in a cell or a tissue are different. The nucleic acids have a relatively low amount of variability, a chemical variability in the molecules present, and are amenable to deep sequencing technologies in genomics and transcriptomics. Whereas the proteins and the small molecules tend to have much higher structural and chemical diversity and are analysed typically using mass spectrometry. So in proteomics, we're using mass spectrometry to enable the identification and relatively quantitate thousands of proteins present in a sample if they're there. And we can do that process in a matter of a few minutes per sample. In the case of a small analysis, we might spend 10 minutes on a particular sample. In a large analysis, or an in-depth analysis, we might spend as long as 3 hours on a single sample. So we do this relative quantitation by measuring the masses of ionized proteins or parts of proteins, and we produce partial sequencing information at up to 100 different parts of proteins per second. And we're able to do that because proteins are composed of amino acids. Amino acids have known compositions, known molecular compositions, atomic compositions. They have a limited range of different elements and their isotopes. Mostly amino acids are made up of carbon, nitrogen, hydrogen, oxygen, sulfur, all of which we know the masses of, and therefore we can calculate the mass of a protein or peptide chain from its amino acid sequence, which we'll dive into a little bit later. So most cell types and tissues produce at least some proteins from the majority of protein coding genes. This means that typically in eukaryotes, in people and plants, we're going to have proteins encoded by upwards of 10,000 genes present in a particular sample. Each of these proteins can be splice variants of a particular gene. They can be post-translationally modified at one or many different sites. So this can include modifications such as phosphorylation, ubiquitination, acetylation, and proteins can be cleaved, cross-linked to other proteins. And all of these different modifications change the mass of the proteins which we see in a mass spectrometer. So this means that we have potentially hundreds of thousands of different proteoforms present in a sample at any given moment. So this creates an extremely complex sample to analyze. And if you wanted to do this manually to quantitate the different proteins present in a sample, it's just not feasible. There's too many different proteins present there. Attempts to analyze different subsets of this diversity have led to a multitude of experimental approaches called proteomics. So by this, I mean that some people are specifically interested in quantitating the phosphorylation status of different proteins. If you're interested in transcription factors or you're interested in signaling proteins, the phosphorylation status of that protein at particular sites is going to be of interest to you because it is indicative of a particular transcription factor being active or a particular signal protein relaying a signal. That's an example of phosphorylation. Things like ubiquitination can be telltale signals that a particular protein population has been tagged for degradation. And so there's a million different modifications, and so different preps and different ways to analyze these samples, but we call it all proteomics. Because of this complexity, we're going to dive into a particular type of proteomics to start off with, because it's probably the basic and most common way to analyze a set of proteins. And that's called bottom-up label-free LC-MS. So we'll unpack those terms and talk about what they mean in the next few slides. So the bottom-up part of that term refers to the fact that we are breaking up these proteins into component parts. It is bottom-up as opposed to intact. In intact proteomics, the heterogeneity of proteins is preserved during the analysis. So they're not cleaved, but they're analyzed as full intact proteins. But because of this heterogeneity that is preserved, it's extremely difficult to analyze many different proteins in a short period of time. At the end of the day, you tend to, excuse me, use intact proteomics to only analyze a few proteins at a given time. So in bottom-up proteomics, we reduce the heterogeneity of the protein population by cleaving it. And this is, the definition here, considering the smaller or less important parts or details of a plan or organization first. So we are breaking up a protein into small parts and then reassembling the proteome from those component parts later in silico, in the data analysis segment of what we're doing. So we're reducing the heterogeneity involves reducing non-peptide backbone bonds. So these are things like disulfide bonds. We reduce the disulfide bonds to reduce the amount of crosslinks that are present in the sample, which means that we have linear amino acid chains left to analyze. And then we are cleaving the proteins up into smaller peptides. Typically, we're looking at peptides which are 5 to 30 amino acids in length. This has a number of benefits. We tend to use a protease such as trypsin to do this. Smaller peptides are more stable during ionization, which we'll talk about later, and it makes them an appropriate mass for analysis in a mass spectrometer. As I said before, this cleaving is typically done with trypsin, which is a proteolytic enzyme. It's active in people. It's secreted in the digestive system. It's inactive when acids are around, so it's inactive in the stomach. And then when the pH becomes more neutral, as food progresses to the small intestine, trypsin becomes active and cleaves up the proteins in food to allow their uptake in the gut. Trypsin cleaves at specific residues. So if you have a protein sequence, trypsin will cleave that protein sequence typically at R and K residues. So the next part of that bottom-up label-free proteomics is the label-free part. Label-free refers to the fact that these proteins that we're analyzing are not chemically labeled in the sample prep process. So it's label-free as opposed to labeled, because in labeled analysis, individual samples are chemically modified with different mass tags. then you can mix multiple samples together and analyze them all together at one time. In label-free analysis, we don't mix the different samples together. Each sample is analyzed individually, and then the data is assembled across multiple mass spectrometry runs. So the LC part of that phrase refers to liquid chromatography. The LCMS part couples liquid chromatography to mass spectrometry. Liquid chromatography, you've probably already come across it. It's an extremely diverse technique. In bottom-up proteomics, we're talking about a particular flavor of liquid chromatography, and that is reverse phase liquid chromatography using C18 as our chromatography media. And typically we do that in a water acetonitrile mix at low pH. So in liquid chromatography, if you're not familiar with it, we are combining the output of two pumps, one of which is pumping water, the other of which is pumping the organic solvent, or acetonitrile in our case, and these flow over a stationary chromatographic medium, C18 in this case, which is, C18 is small silica particles. The media that we use in our facility is about 1.9 micron average diameter of particle, and on those silica particles, or sand particles, there are carbon chains which are 18 carbons long. Those carbon chains provide a hydrophobic surface for proteins and peptides to bind to in specific conditions, which we'll go into in the next slides. So as I said before, proteins have diverse chemical properties, in part due to the amino acid side chains that they're made up of. Some of amino acid side chains are hydrophobic. These are typically long side chains, long as in they have extended carbon chains, and the alternative is hydrophilic amino acids, which have short or hydrophilic side chains. So when cleaved, the parts of proteins or peptides have characteristic hydrophobicity, and that hydrophobicity is a result of the constituent amino acids that peptide is made up of. In reversed phase LC, we use a hydrophobic stationary phase, C18, and we introduce the peptides in a 2-aqueous phase. So typically, we introduce the peptides to the chromatography media in 2% acetonitrile, so it's 98% water. In this case, all of the peptides basically are too hydrophobic to be happy in the water, and they will by preference stick to the chromatography medium. So initially, all peptides stick to the stationary phase. In the LC part of LCMS, we then increase the proportion of organic solvents in the mobile phase linearly. So the liquid chromatograph, which we'll talk about more in the next lecture, which is initially pumping 2% water, 98% acetonitrile. We then, over the course of the analysis of the sample, increase the proportion of acetonitrile which has been pumped in a linear manner from that 2% up to something like 30%, maybe 35%. Over the course of that linear increase in acetonitrile concentration, each different peptide chain at a specific acetonitrile concentration loses its affinity for that hydrophobic C18 medium, and then falls off or comes off the stationary phase and is eluted out into the mass spectrometer. So we do that in a column, in a fused silica column like these ones, and as that acetonitrile concentration increases, the most hydrophilic peptides, the ones composed of amino acids with the shortest or most hydrophilic side chains, come off in the early parts of that gradient at low acetonitrile concentrations, while the higher, more hydrophobic amino acid, longer side chain containing peptides come off later in that gradient. You know, in an ideal situation, if you had a low complexity sample, hopefully each different peptide is eluting at a different time in that acetonitrile gradient and is introduced to the mass spectrometer alone or solo. in reality, because we have those hundreds of thousands of different proteoforms present in a sample, we're always going to have many, hundreds, maybe thousands of different peptides eluting off at the same moment in time. The chromatography doesn't have the resolving power to completely baseline separate all of those different peptides. So over the course of that LC gradient, the liquid chromatography gradient, the MS operates continuously, is ionizing the output of that LC gradient and flipping back and forth between two different modes of operation. These are MS scans and MSMS. The MS scan mode refers to an uncollided or an unbiased sampling of everything that's coming off the LC at a given moment in time. So it does that for a complete mass range every second if we give the mass spectrometer that duty cycle, and it will sample all of the different mass charge ratios, or all the different masses of peptides that are coming off at a given hydrophobicity. And it will give us both the mass charge ratio of the analytes which are coming off the gradient at that point in time, and it will give the intensity of each of those different analytes. So this plot you see in the bottom right corner here is one MS scan. So these, I'll point out the axis labels here. On the bottom axis we've got mass charge, or mass, from low mass to high mass. And we've got the intensity on the z-axis, on the y-axis, sorry. So different analytes, if the protein is higher in abundance, roughly speaking, there's caveats here, I'll get to them later, the intensity of that peak will be higher in an MS scan. But an MS scan doesn't tell us what is present in the sample. It doesn't give us the identity of the amino acids. There's too many different peptides with overlapping masses for the mass charge alone to give us the identity of that peptide. So in order to supplement that information, we're constantly doing MSMS at the same time. So the MSMS refers to an MS scan on a particular part of an MS scan. In the case of proteomics, we are identifying a specific analyte in the initial MS scan, a particular mass. We're then crashing that particular mass into gas molecules to start to break it apart. And then we are collecting the fragments that collision produces, and then we are getting an MS scan on those fragments. We'll talk more about that in the second lecture. So in terms of MS scans, as I said, the mass spectrometer is operating continuously throughout the gradient, LC gradient. It is collecting MS scans. I try to make it collect MS scans at least once every second. So in the case of that, for a particular mass now, for a particular M over Z, we can start to assemble an MS feature. So chromatography is not perfect. All of the peptides which are present in a particular population do not come out at a specific point in time. They come out in a elution curve. So a little bit of that peptide will come off early, a little bit of that peptide will come off late, most of it will come out at its given hydrophobicity. So we need to assemble over time that elution gradient to give us something to compare the abundance of between samples. So here we've got multiple MS scans taken at different points in time here, which we can then integrate over time to turn into an MS1 feature or an MS scan feature. When we look at an analysis software downstream after we've finished the analysis of the sample, we see the evidence of those individual MS scans. You can see that there's a jump up between these 2 points in time, another jump up here, here, here. So what we've done is draw point to point between the MS scans to give us a peak that we can then integrate to get an area under the curve measurement to compare between samples for a particular peptide. So in this analysis software, obviously we've got an MS scan, we've got the analysis of a particular peptide over time in this window, but we also have the MSMS scan, which allows us to identify that peptide in this window here. So we'll see that this window has a series of different Y ions. So this is mass charge ratio and intensity for a particular mass after it's been collided at a particular moment in time in the mass spec gradient. And I'll try to show you some more examples of data that might start to make that feel a little bit more intuitive. But for now, a Y ion series is what we've used to identify this amino acid chain. And the Y ion series is a partial sequence for that peptide chain. So this is the same MSMS you saw on the previous slide. We've zoomed in on it here to look at the match. I can see now that we have made a match for this particular MSMS spectra. And that sequence has come out as an amino acid sequence that we see here from N to C terminal. So how that happens is that when you collide a peptide, a sequence of amino acids into gas molecules, you tend to get fragmentation along the peptide backbone. So individual amino acids will fall off and from either one end or the other end, basically. So here I've tried to show that in this peptide, QILYLISK, we've got 1, 2, 3, 4, 5, 6, 7, 8 different amino acids in this chain. We haven't seen that entire chain here, but we've seen Y6, Y5, Y4, and Y3. So we're talking about, where are we? LYLISK. This part of the sequence is our Y6 ion. The difference between the Y6 ion and the Y5 ion is that we've knocked that initial leucine off. The difference between the Y5 and the Y4 ion is that we've knocked that Y off. And we can tell that that's occurred because of these characteristic mass shifts between the Y6 and the Y5. Between Y6 and Y5, we've got a mass shift of 113.1. And if we look through our masses of different amino acids, we'll see that both isoleucine and leucine have that typical mass shift. And there are two amino acids that have the same mass shift because leucine and isoleucine have the same atomic composition. They just have a different configuration. Whereas we see that the mass shift for the Y, as we knock that off, is larger because our Y amino acid, tyrosine, has a different a different atomic composition, and that has resulted in a bigger mass shift. So I've mentioned the concept of B and Y ion series. These basically are just taking that same peptide that you see here and either knocking amino acids off from this end, from the N-terminus end, or knocking amino acids off from the C-terminal from this end. So the B is from the front, the Y is from the back. So there are two different types of data that we've collected in a mass spectrometer. We've done it continuously over an LC gradient, but what does that get us? Integrating the intensity information from a specific mass charge ratio, a specific mass, from MS scans over time, as I mentioned, gives you an area under the curve measurement, which you can compare between samples. And then the MS, or fragmentation spectra, can tell you what the amino acid sequence of that target was. So now we know what the sequence of the MS1 feature was, and then we can compare the size or the intensity of that MS1 feature between different samples. So in the example that I've brought up here, we've got the area under the curve, or the peak area, for a series of different samples here that are collected. In this case, we grew up wheat leaves or wheat plants under salt or non-saline conditions, and then we chopped the leaf up into different sections, and then did this in triplicate and analysed each of those different sections to see what the abundance of several thousand different proteins over the course of the analysis was. This particular peptide from a particular protein was not present in the control sample. We can see here the consamples, the control samples, all have zero abundance. But we see that this particular protein increased in abundance in the salt samples over the leaf gradient, basically. So from base to tip, there was an increasing amount of this protein. So this can tell us about the metabolic state of that particular wheat plant and how that metabolic state evolved over time. And we can theorize about the function of this protein from the way it has responded to the salt treatment. So let me just spring out of the presentation for a second and we'll have a look at this in a different way that I find more intuitive. So this is a single mass spectrometry run. So this is a single sample. And if we look at the x-axis here, we've got the masses of the different analytes that were present in that sample. If we look at the y-axis, we have time. So this is really a three-dimensional set of data that we're looking at. We've got mass, time, and then in intensity is color-coded in this set. So if we have a look closely at one of these pieces of data in this set, something like this is going to be an individual peptide from an individual protein. And as you can see in a zoomed in look at this data, we can see that there's 20 or 30 different peptides that we can see here, all at specific masses, and then they have a characteristic retention time which they smear out over a little bit. But as we zoom out from a particular peptide, we can see that we have just thousands and thousands and thousands of different peptides in this sample. At different hydrophobicities, in this case, over time, the more hydrophilic peptides are going to be coming out early in the gradient, and the more hydrophobic peptides are coming out later in the gradient. So if we zoom back in again, we can see that some of these peptides are more intense. They have more red, purple colors. Some of them are less intense. They have more green and blue colors. And that is related, although not directly, to the amount of protein or peptide that was present in that sample. So if we click on some of these other buttons here, we will get all these purple lines. So what these purple lines represent is this particular MSMS event. So these features, as I've mentioned, are assembled of a series of MS scans over time across the peptide elution curve. And as that has occurred, the mass spectrometer has identified that that is an interesting looking analyte. It has a series of algorithms that it uses to identify interesting looking analytes. In this case, we've told it to look for things that look like peptides. That algorithm has then identified that this isotopic series or analyte looks like a peptide, and then it has specifically isolated that peptide for MSMS. And so each purple line that you see in this plot is one MSMS event. And what's occurred is that the mass spectrometer has seen that mass in an MS scan. It has then identified that it's an interesting looking mass. It has then isolated that particular mass. It has then collided that mass into gas molecules. It's then collected the fragments from that collision, and then it has looked at the masses of those fragments. So that gives you MS, MS, and that can give you, over different samples, a relative quantitation of the amount of peptide or protein that is present in different samples. So I've just given you the summary, but here again in slide form, the proteomics speedrun summary is that bottom-up label-free LC-MS allows the identification and relative quantification of tens of thousands of peptides and thousands of proteins in acceptable timeframes. This is done by collecting MS scans, which contain information on the number of ions present at a given mass at a given time. And this info is used to identify targets for MS collection, which provides partial or complete sequencing of the target. The abundance of the target can then be compared between samples by matching the sequencing data to the MS scan data to give an intensity per sample. Hopefully that's clear. If it's not, please ask questions. As I said, that's a speed run. There's a lot more details in making good decisions about proteomics and mass spectrometry data. And when I say good decisions, I mean what can you conclude from that data, and how do you design experiments that can leverage this data to access your biological questions. So ionization is something I've skipped over in that process, but critical to mass spectrometry. Mass spectrometers don't analyze the output of the liquid output of chromatography or chromatographs. They need to first ionize those outputs. That process is called electrospray ionization, usually for proteomics. There are other types of ionization. When I'm analyzing samples, I exclusively use electrospray ionization because it gives you the ability to get the deepest into samples, identify the most proteins and get the most quantitation of different samples, of different proteins in a particular sample. So in electrospray ionization, we are bringing the output of a liquid chromatograph down to a very fine point and spraying that liquid out into the ion source of a mass spectrometer. That fine spray contains acid because we put it into our mobile phases. It contains a mixture of the different peptides that are coming off the column at that moment in time, and it contains the mobile phases themselves. In electrospray ionization, those liquid droplets are sprayed out into a high differential voltage between the tip of the emitter, which comes from the liquid chromatograph, and the entrance to the mass spectrometer. That voltage differential, along with drying gas, causes the liquid droplets to rapidly desolvate, we call it, is basically evaporate. And in that process, in the high voltage, the peptide chains take on those hydrogens that are there from the acid present in the mobile phase and become ionized. So we go from liquid phase peptide mixtures into vapor phase, ionized, and now magnetic peptides. They're magnetic because they're charged, they're ionized. And we use that magnetic property or electromagnetism of the peptides in the mass spectrometer to perform the slightly magical things that mass spectrometers can do. Mass spectrometers can use mass filters to filter different masses or mass over charge ratios, and we'll talk about them, or actually we should talk about them now in the ionization. So mass spectrometers, they measure masses, but in fact they don't measure masses at all. They measure mass to charge ratios. They measure the ratio of the mass to the amount of charge that a particular analyte is carrying. So if you have a peptide that has a +2 charge, which is common for peptides because they have 2 primary amines when cleaved by trypsin, the mass charge ratio of that thing will be different to what it is when it's at +3 charge state, because it is the mass divided by the charge. It's whatever mass it was divided by 2, or whatever mass it was divided by 3. An important consideration about ionization and an important limitation about mass spectrometry and proteomics is that not all peptides have the same propensity to form ions. Some peptides will readily ionize, and then we'll see them brightly in the mass spectrometer in high intensities, and other peptides will be reluctant to ionize. So for every molecule of peptide, you will get less ions formed. And what that does is break the direct relationship between the abundance of a protein in a sample or a peptide in a sample and its intensity in a mass spectrometer. And that's an important consideration for analyzing the data downstream. So ESI is about transferring proteins as ions into the gas phase without breaking them, importantly, so that we can break them later when we perform MSMS scans by crashing them into gas molecules. So we've talked about the breaking of a peptide backbone at different amino acids and how we can use that information to build up a partial sequence for a peptide or an ion. Doing that manually is going to be very painful if you're trying to do that for 50,000 different analytes in a particular sample, and then you want to do it across 24 samples. In some sample sets, we have 10,000 samples in a set. So it's completely infeasible to do that manually. And what we do computationally is that in high throughput in an automated fashion. And we do that by assembling all of the protein sequences from a particular genome of a sample. If I know that I'm analyzing wheat, I get the wheat genome, and I find the coding sequences for all of the genes in the wheat genome, and I translate them to proteins in silico, and then I use that as the input to a trypsin cleavage algorithm. So what that does is look at all of the protein sequences, find all the R and K residues, and cleave them up into the theoretical peptide fragments that all of those proteins would make. So what we can do with that is use the known amino acid sequences that a particular genome or proteome would produce to make a list of all of the theoretical precursor masses that could exist in a sample. From the MS data, we know that what the mass of each MSMS spectra was in the MS level, so that what the mass of the peptide backbone was. And we know the precursor, So what we do then is when we have our list of theoretical masses is that we can put a bracket around our known mass of our analyte and compare the MSMS spectra to theoretically produced MSMS spectra from all of the nearly matching masses to that particular peptide mass. What that allows us to do then is calculate correlation coefficients between the MSMS spectra, the B and the Y ion series that we measured in the sample, and calculate the correlation coefficient between all of the close matching masses in our database. We can then pick the best, or the highest correlation coefficient between those candidate amino acid sequences, and that gives us a probable match at that point between our MSMS and a protein that produced it. So I've got winner takes all, the best one wins out of that correlation coefficient calculation exercise. So you can download these peptide FASTA files from resources such as UniProt, which in the case of the E. coli genome or proteome, there's about 4,400 different protein sequences that you can download in a single click into a FASTA file. So that's how you would access that information. So here we're just diving back into what I've just described on the previous slide. We've got an MSMS spectra. We know the precursor mass, what the mass of that analyte was before it was broken up. It was 337.2264 m over z. So we go into our theoretically created list of masses, And then we put a mass tolerance around that specific mass. In this case, we've got plus minus 0.002, and we've identified 4 different amino acid chains that have masses that nearly match that mass. We say nearly match because mass spectrometers are not completely accurate. They have a tolerance range, so you can't say from even a carefully measured mass exactly what it was. So we bracket a window around that and compare the correlation coefficients between, you know, those 4 different matching masses. In this case, I've got 4 different peptides here, all with the different Y ion series. In this case, and we seem to have lost the graphs. We can see it in the non-presentation here. I've just mapped out, if we were comparing the theoretical Y1 the measured Y1 to the theoretical Y1 for this particular peptide. We put it there, Y2, Y3, Y4, Y5, Y6. And we can see that that is quite a scatter because this is different amino acids in this peptide to the ones that were really present in the sample or measured. And we can see we get poor correlation coefficients for the other 3, but really good correlation coefficient for the one that matches. In practice, we don't calculate correlation coefficients because it's computationally inefficient, and we want to do this on large scales. We are just doing dot products, so we're multiplying the 2 vectors together. So this protein identification or matching in bottom-up proteomics between peptides and proteins is not straightforward. When we cleave proteins, we generate a set of peptides, and it is the case in most complex genomes that many of these peptides will overlap between different proteins. This can be for multiple different reasons. So different protein isoforms, if you have multiple copies of the same gene, then they may be producing isoforms which are very similar, protein isoforms which are very similar in amino acid sequence or identical in amino acid sequence, but are actually derived from different genes. You also have functional domains. These are often have very similar amino acid sequences, in which case, all of the different TOM20s, for example, would have very similar active sites or functional domains in them, which we may not be able to distinguish between after they've been digested by trypsin and matched through this database algorithm. And also by random chance, you'll get proteins that have matching amino acid sequences in part of them. So where these overlap, the bottom-up approach isn't actually able to determine which protein they arose from. So protein assignment is imperfect and is done on a best evidence basis. So what this means is that in conservative protein lists that we generate from the 10,000 different peptide IDs we might generate from a particular sample, we assemble a minimal set of proteins usually. So this is the minimal set of proteins that can explain the peptides identified, i.e. if you have a peptide which is matched through MSMS in your sample, but it maps to multiple different proteins in the proteome, that is not necessarily good evidence, right? If there are other unique peptide sequences, i.e. other peptide sequences that don't occur elsewhere in the proteome, for some of those proteins, that's going to contribute stronger evidence that that protein was actually present in your sample than if you don't have that evidence, i.e. you've only got a redundant peptide sequence that occurs in multiple proteins. That would be weak evidence that that particular protein was present, particularly if other proteins have unique sequences. So I've tried to break this down for you as to what this looks like. I've been talking about wheat a little bit because I've done a lot of work in wheat historically. The challenge with the wheat genome or proteome is that it's hexaploid. It has 3 copies of every gene basically, because it was hybridized multiple times in its evolution. This means that you're going to have 3 very similar sequence proteins for every gene. And we see this here for pyruvate dehydrogenase E3 subunit. We've actually got not 3, but we probably had 2 isoforms in the initial before hybridization genomes, which has resulted in 6 obvious homologs or obviously related isoforms of this protein, and 1 less related protein, possibly a cleavage product, a truncation of one of those genes. But we see that around each of these genes in blue circles, I've mapped the peptides. In green are peptides that we identified in the process of analysing this sample. And we can see that some of these peptides, like this one here, can be derived from, I think, every different gene, right? So they're in common to all 7 different proteins that PDHE3 could be in wheat. And we see that some of these are only related to a single protein isoform. So this would be unique and strong evidence that this particular isoform is present. This would be weak evidence that any one of these isoforms is present, though it is strong evidence that the gene family is present, right? So there's it tells you that PDHE3 is there, but it doesn't necessarily tell you exactly which gene contributed that PDHE3 protein. So a few more details about are important for the understanding of proteomics. Here I've got an MS scan, particular moment in time from a sample analysis, intensity on this axis, mass charge on this axis. And if we zoom into this particular point of this MS scan, we see that those peptides, and we saw it before in BATMAS2, that an individual peptide is not actually a single mass peak. It doesn't occur like that in nature, which is slightly disappointing from the mass spectrometry perspective. But in reality, the carbon that is present in the environment is not all carbon-12. About 1% of the carbon that's present in the environment is carbon-13. This is somewhat distinguished against in photosynthesis, but there's plenty of carbon-13 present in plants, and that makes its way into proteins, into amino acids and proteins through metabolism, and it does so in non-defined ways. And by that, it'll mean that when we look at the isotopolog series of a particular peptide in a mass spectrometer, there are likely to be some carbon-13s present in that peptide population, but those carbon-13s are going to be present at different points or in different amino acids in that carbon chain. So carbon's not the only one that has stable isotopes present in the environment, but it's the major one. 1% of carbon present is much more than is, the 0.0001% of deuterium, which is present in the environment, or heavy hydrogen, H2. The same for N15, heavy oxygen and sulfur. So that means that when we're dealing with biological mass spectrometry in non-isotope enriched fractions, that most of these isotopologs that we see are going to be carbon-13s. So this isotopolog series has a beneficial offshoot because it allows us to determine the charge state of that particular analyte. The charge state is important because we want to know what the neutral mass of a peptide was, right? Independent of M over Z of the ionization process, we're going to want to know what the specific mass of that amino acid chain was. If we didn't have this isotopolog series, we wouldn't necessarily know what charge state the thing was. And it tells us the charge state in combination with the presence of heavy carbon because we see a characteristic spacing between the peaks. So this 876.9506 is the monoisotopic mass. So it's all carbon 12s in this case. But we see that right next to this, we see 877.4514, and then we see 877.9525, etc., etc. And if we do the maths, we see that these masses are incrementing by almost precisely 0.5 each time. So that's what makes an isotope log series. And that's because this is a +2 charge state analyte. And we can tell that because we know that these are going to be carbon 13s. There's going to be one carbon 13 different between the monoisotope and the +1. So this means +1 carbon isotope. The +2 carbon isotopes present, +3 carbon isotopes present, +4 carbon isotopes present. So as the length of the amino acid chain goes up and the number of carbons that are present in that amino acid chain increases, so does the proportion of those peptides which are going to have a carbon 13 present in them. So, you know, as you approach 100 carbon atoms, most of the peptide chains at that point are going to have at least one carbon 13 in them. A bunch of them are going to have 2, a bunch of them are going to have 3 or 4. So when we look at that spacing between carbon 12 and carbon 13, as I think I've probably belabored now, we know that there's 1 AMU difference between those different peaks. And when we do our m/z calculation, our mass over the charge, that 0.5 tells us that this is a +2 because we've got a 1 difference and we've divided it by 2, which gives us 0.5. And likewise, if you had a +3 charged analyte in the mass spectrometer, you would get a 0.33 recurring mass difference between the carbon isotope log peaks. So this is just highlighting that. This is a peptide, which I've looked at previously. If we double its length, i.e. we just put 2 peptides together, maybe we missed the trypsin cleavage, we double the number of carbons that are present in that amino acid chain, and it modifies the shape of that isotope log envelope, which we'll talk about more and how you can use that in lecture 3. So, but there's more. As I said, when you have multiple stable isotopes present in the environment, you have 1 neutron difference between both a carbon 13 and a nitrogen 15 as opposed to an N14. Both have 1 neutron difference, but the mass difference between these 2 different isotopes is not the same, because E equals mc squared, and you get a difference in the binding energy of that particular isotope. Or you get a difference in the binding energy of that isotope. So that means that our +1 peak and our +2 peak in reality are made up of multiple different masses. But generally in mass spectrometry and proteomics, you don't need to worry about that too much. But we can use it, and I will talk about that a little bit later on. So I talked about, just briefly touched on labeling and label-free proteomics in a previous slide. I'll just unpack labeling for you a little bit because it is common. You'll come across it all the time in the literature. And it can be the best way to do proteomics in certain circumstances. What labeling refers to is a chemical modification of the peptides post trypsin digest in your sample. So I think the current maximum that you can get is an 18-plex reaction. That means that you can analyze up to 18 different samples by labeling them with different masses. So what that means is that you're going to isolate and digest your proteins, 18 different samples at this point, and then you're going to encode each one of those with a different mass. We call it a reporter. You're then going to mix all those different samples together with their different mass labels, and analyze them together in a single mass spectrometry run, single LC gradient. So what's clever about this approach is that although you're adding different masses, you're adding the same mass, because each of the different mass tags that you add to a sample consists of a mass reporter and a mass normalizer, which is then linked to your sample peptides. That means that when the peptides from all your different samples are mixed together, they can enter the mass spectrometer all together. But when they get to the MSMS portion of the mass spectrometer, so when they're collided into a gas molecule, the different mass reporters are knocked off. And so this allows you to look at a telltale mass in your MSMS, which gives you a quantitation or relative quantitation of how much of that particular peptide was present in each of the different samples by looking at the different mass reporter. I hope that makes sense. Please ask questions if it doesn't. So all of the mass reporters weigh the same until you break them apart, and then the presence of different carbon isotopes, heavy isotopes, between the mass reporter and the mass normalizer allow you to distinguish between the samples. This has advantages because it means that your samples are more comparable. You can get a lower relative standard deviation in a quantitation using this technique than you can in a label-free. It's not a huge difference, but it is a real difference, and it can be important. The disadvantages are that it costs money. It's real money. It costs thousands of dollars to, I'm not sure what the cost on an 18-plex sample prep kit is, but it's many thousands of dollars. It also limits the maximum number of samples that you can analyze together without doing complicated normalization between runs. So currently that maximum is 18 samples. If you've got want to analyze 24 samples together, this may or may not be the ideal technique. It also suffers from ratio compression. So when we do that, taking all of the peptides together through to MSMS, if you have one sample which is particularly strong in that particular peptide, there's a lot of that peptide present in one sample and substantially less of that peptide present in another sample, it will tend to, the mass spectrometer will lack the dynamic range to make an accurate comparison between those two things. So particularly if you're comparing really dissimilar samples where you might have none of that protein present in one sample and a lot of that protein present in another, you will get a smaller ratio between those reporters than you would expect, than is reality. But one of the big advantages of this approach is that it's really amenable to fractionation, which we will talk about in a later lecture. So we've talked a little bit about how the direct connection between the intensity of an MS1 feature and the amount of that peptide or protein present in a sample is broken, but through the ionization process. So it is really useful to be able to say what proportion of a sample was a particular protein, and what ratio different proteins, perhaps that are present in a common metabolic pathway, are present in a sample. You might want to compare, the abundance of succinate dehydrogenase to ATP synthase. In that case, we use something called a normalized spectral abundance factor or abundance factors. What these do is just process the data basically by dividing the number of peptides that you've identified that belong to a particular protein by the length of that protein. So that tries to create a common ground so that you can compare, a big protein that might have 500 different 500 different peptides, it'd be a really big protein in that case, 500 different peptides to a small protein that only had 10 different peptides. that's not an equal comparison basis, but when you divide the number of peptides identified by the length, hopefully you start to reduce the influence of that difference. And so you can use that normalization process to get an estimate of the percentage of a sample which was comprised of a particular protein. In practice, I talk about them because they're very common in the literature, they're very commonly used, it's the right way to do it, but in practice these abundance factors are not really much better than just saying what percentage of MSMS IDs belong to a particular protein in your sample. Dividing by the length of the protein is not that useful. So another important consideration when you're designing mass spectrometry experiments is ion suppression. It is unavoidable that other analytes are going to be present in your sample, and unfortunately when you are ionizing multiple analytes together, if one of those analytes is high in abundance and one of those analytes is low in abundance, the high abundance analyte tends to crowd out the low abundance analytes. So I've tried to show that here. I don't know if it's super obvious, but you can see that this is sorry, I haven't put the axes on this graph, but this is mass to charge ratio on this axis and this is time on this axis. We can see here that in parts of this elution gradient, I can see that the background or the amount of blueness in between the peptide samples has reduced. And that's because we've got a series of really high abundance peptides which are ionizing at that particular moment. So what that's done is crowded out the lower abundance and noise present in the sample, because the mass spectrometer only has a limited dynamic range and a limited ability to ionize ions. If you've got too many of one type, you can't see all the rest of them effectively. And so that has really important implications when you are comparing dissimilar sample types. We refer to it as the background matrix of a sample. If you say if you specifically isolate one particular protein out of a sample, i.e. you do a tap tag or an affinity purification for that protein, you enrich it up until it's millions of times more abundant than anything else in that sample. That background matrix now no longer matches the sample that it came out of. So you could not compare the abundance of that protein between those two samples, because in the complex sample now you have multiple analytes that are coming out at the same time as those pure peptides from that particular protein, and they will change the response of the mass spectrometer to the abundance of that peptide in the output. So what I was saying, I hope that's clear. If it's not, please ask questions. Analytes crowd each other out. So very abundant ions will push down the signal of the less abundant ones, which means that the best, most quantitative experiments are going to be ones where the sample background is very much the same. Maybe you're comparing two different leaves, and you've got few proteins that are changing in abundance against that background. So I've touched on dynamic range. Dynamic range is a huge consideration in mass spectrometry because the dynamic range of mass spectrometers is smaller than the dynamic range, the real dynamic range of protein abundances in nature. This is a figure pulled from a paper. It's an old paper, 2004, I think, where they looked at the dynamic range of proteins best as they could in human blood plasma. And they see a greater than 10 to the 10 range. So that basically means that, some molecules you've got 1 of, other molecules you've got 10 to the 10 of, which is just a staggering dynamic range. And an unfortunate side effect of this large dynamic range is that in mass spectrometry we will rarely or never analyze proteins. We'll never identify proteins that are right down the bottom end of that dynamic range. If you want to identify a specific transcription factor that there might only be 2 or 3 of in a cell, you know, that's going to be a very low abundance protein compared to something like Rubisco, the most abundant protein in the world. You're going to need to do a tap tag or have some means to increase the proportion of protein in a particular sample that transcription factor comprises. Otherwise, it's just going to be squashed. You're not even going to see it. It's going to be down there in the noise floor of the mass spectrometer. And so this dynamic range issue also means that the more abundant proteins will commonly have more signal intensity in the sample. And that has some offshoots. In mass spectrometry, the brightest signals take the least time to analyze. We'll talk more about the functioning of mass spectrometers in the second lecture, but suffice it to say that analyzing different abundances takes different amounts of time. It's faster to do it for high abundance molecules. That means that you can identify more peptides of different sequence in a shorter period of time if they're high abundance. So we usually specifically go for really high abundance peptides first because it increases the total amount of proteins that we identify in a sample. So in this slide, I'm trying to demonstrate that typically when we're doing MS scans, we see only a portion of the sample. We see the brightest and the most abundant portion of that sample, but that doesn't mean that the other things are not present. On the right-hand pane here, we have a standard MS scan acquisition of a particular sample. But we can do better than that by using the tricks of the mass spectrometer and the way we use it to dive deeper into that sample. So we can see that when we do something called a boxcar, you can look that up. It's basically using a mass filter to increase the amount of a particular mass range that you see in a sample. When we do that, we can see that like these guys here are present in this sample. This guy here present in the sample, but we can start to see that there are many more analytes actually present below the limit of detection of the mass spectrometer when it's collecting MS scans. So know that, you know, a mass spectrometer is not perfect. It's always going to miss things, and it's going to have an abundance biased sampling of your sample. I think we're getting towards the end here. Resolution is something you'll also see talked about in mass spectrometry. Not all mass spectrometers are created equal. Some mass spectrometers cost $1 million, some cost $2 million, others cost less than $100,000 because multiple reasons, but one of them is resolution. That resolution refers to the mass charge dimension of a mass spectrometer. When you have a low-res instrument, its ability to differentiate masses is less. So resolution is the width of the mass peak that you see. So now we're talking about M over Z on this dimension. This is not what has it been in previous times. It's time in the chromatography dimension. Now we're talking about mass charge dimension, and this is intensity on this axis. So we actually see that the mass spectrometer is not perfectly capturing a single mass. We're actually getting a spread of masses for this. In low resolution instruments, that spread is wider than it is in high resolution instruments. So you can see here we've got one Dalton from here to here and one Dalton from here to here. So this peak is about 14 times narrower than this peak. So the resolving power of this mass spectrometer is 14 times higher than this one. This is not a particularly high resolving power mass spectrometer. Resolution here is 14,500, typical of QTOF instruments. Good Orbitraps often go to 500,000. The best Orbitraps can be over 1 million in their resolution. Just finishing up, often resolution is related to mass. Depending on the type of instrument you've got, some of them will have higher resolution, higher in the mass range. Some will have higher resolution, lower in the mass range. It's not a constant across the mass range. And commonly, we throw away data relating to the resolution of the instrument as we're collecting data. We call this peak picking or centroiding data. Generally, if we've collected this MS spectra, I think as you've seen previously, there's no curve to this data. What we do is we pick the maximum intensity of that peak, in this case 613.9642 m/z, and we only store that m/z along with its intensity and throw away the rest of the data. And what that does is give us smaller file sizes and slightly easier data processing downstream, but it may limit your ability to use your data downstream if you don't pick up that data. Max quant, which Elke will talk to you later in the lecture series about later, requires that data not to be centroided. So you need to be aware of what you're doing to your data in the process of collecting it. So this slide, I think is going to be the last one. We're just talking about different axes on graphs. This is the bat mass thing that we saw before for a particular peptide. Often we're referring to mass charge ratio on the x-axis and retention time on the y-axis, but sometimes, as you've just seen in the previous slide, we might be referring to mass charge on the x-axis and intensity on the y-axis. And so these graphs can look very similar, whether it's chromatography or mass. Just be aware that there's complexity there. So I'll leave it at that for today. Thanks very much for your time.